All right, we are recording, so I'll hand the floor over to Group 10 to share with us their bioreactor design.
All right. Well, firstly, we just wanted to say thank you to everyone for coming out. We're Group 10, consisting of Jenna Agello, Haley Fritz, Tyler Lee, Ryan Oliver, Locke Tran, Alec Weaver, and myself, Brooke Towns. Our design is called the SABRE, which stands for Semi-Autonomous Bioreactor. I'm really excited to share with you what we've been working on throughout the whole semester. So, there we go. So a quick outline of how this presentation is going to go. We're going to go into our Hedgehog concept and a brief product overview. Then we're going to break it down by subsystem so you can really understand exactly how the SABRE works. Then we'll talk about competitive advantages, not only over other teams in EML4501, but products currently available on the market. And then we'll break down the cost, offer some conclusions, and then we'll accept questions at the end. So the Hedgehog concept that our group decided to pursue this semester was to offer the most autonomous machine possible. Now, obviously, due to time and budget constraints, we can't fully realize a completely autonomous machine. However, the SABRE does offer automated liquid handling between our custom culture plates, which we will go through in a few slides, and any commercially available well plate or conical tube, as well as self-sanitizing via UVC lights and an ethanol solution, which isn't currently available on the market either. So if you'll look on the right, there's a picture of how one could use SABRE with a typical vent hood and a woman for scale. Some key features of the design are the dispensing needle and micro pump for fluid handling, the customizable well plate, the ability to offer orbital, double orbital, and linear shaking patterns, and the ability to take optical density and fluorescence intensity readings of individual wells. The overall size of SABRE is 48 inches long, 24 inches deep, and 24 inches tall, and the weight is about 175 pounds. And at maximum, the power consumption would be 750 watts, but that would be a conservative estimate of the actual maximum, which wouldn't typically be used. We broke SABRE up into 6 subsystems, including the sensing and imaging, heating and cooling, dynamics and translation, microbiology, liquid handling, and the overall structure. And then shaking plates are also noted individually on this slide just because of their distance from the rest of the dynamic system, but they are included with that as well. And with that, we will dive into liquid.
Hi, so this is our liquid handling portion of the design. It functions to dispense liquid into the well plates, and it also disposes and neutralizes the resulting waste. It is also capable of disinfection so that there is no cross-contamination when the needle moves from each well plate. It is at a technology readiness level of 3 because the concepts have only so far been demonstrated analytically and not yet in a laboratory environment. However, the OTS parts for this design are at a technology readiness level of 9. Next slide, please. This is an exploded view of the liquid handling subsystem. It shows the different components from the liquid reservoir to the stainless steel dispensing needle. The stainless steel dispensing needle can dispense the liquid into the well plates and also bring it back up once it's waste. The waste container is also located here, as well as the 70% ethanol solution. And there are soft tubings that connect to the micro pump and also connect to the dispenser body. The body is then connected to the dynamic system, and it is composed of nylon because this material is resistant to chemicals and is also strong and stiff, containing good wear properties. This table here outlines the customer needs that are met by the liquid handling system, such as the power to run, operational lifetime, and product cost. Some requirements that only this subsystem fulfills is a dispense rate of 300 microliters per second, met by the customer requirements, a maximum and a minimum customer desired volume of liquid and the ability to dispose and neutralize waste. The maximum dispense rate that the liquid subsystem has is 670 microliters per second. The total cost of this product is also 11.4% of the $10,000 budget. And now Haley will talk about the microbiological system.
Yes, so for the microbiological system, we have customized acrylic well plates. So there are 384 of these square containers that are made out of acrylic, and these are going to be injection molded, which allows it for just easy manufacturing since there are so many of them. And then enclosed on them are the sandwich plate lids. So these sandwich plate lids are made out of 3 different layers. There's a microfiber filter inlay layer, and there is a PTFE filter and a silicone layer. So these layers allow for the liquid disposing needle to go through the top, but allow for fluid and gas to not go through at all, which is good for the translation system. And this is also another example of this would be a basketball when you're pumping up air. That's basically how it works. Also with them are the 5 solenoid valves and a flow meter to control the gas flow. For the TRL for this microbiome system, it's a level 2 because this is a unique design and it has not been prototyped yet. So this is the whole outline of the microbiome system. So I'm just going to start giving a layout and a rundown of how everything is processed. So it starts at the flow adjustment valves, and these valves are just used to manually turn them on and off. Since we're using such highly reactive gases, you want to make sure we know which ones are on and which ones are off. And this just is an easy way to do it. Going through there's solenoid valves, and these solenoid valves are electronically controlled, and these allow for electromagnetically coils to plunge the system and to turn off the valves or turn on the valves. And then going through there is a flow meter, and this flow meter shows the mass flow rate of the gas, and there is a user interface on it which allows to show the gas flow rate, and it also has an alarm which will turn off which will be notified if there is a problem. And then going through there, it goes through one port into the system, which is the sandwich plate lid, the customized well containers, and these customized well containers on the side have pipes, and on the pipes, these plastic pipes, they are sealed through an O-ring, and each one individually has their own pipe. That way it is an enclosed environment and has the gas through each individualized container. And through this, there's a container plate with holes for the pipes to go through, and then there's just a bottom tray for the total pipes to go through so that it can go through the singular port to link up. And also, once they are done and we want to eject all the gas, you would just lift up the sandwich plate lid, have the structure lid open, and then we would use the hood vent. And to purge out the system, we would use carbon dioxide and the heating and cooling fans to basically clear out the system. The carbon dioxide the carbon dioxide is so little that it won't affect anywhere for any of the lab participants. So for the microbiome customer needs, there are 7 requirements, one of which is the cost, which is 20% of the budget. And another one that is important is capable of incubation, which is shown through the next slides through heating and cooling.
All right. Hello. For our heating and cooling, we decided to use thermoelectric coolers, which will be off-the-shelf parts that consist of 2 ceramic plates and NMP type semiconductor junctions, in which a current will be ran through them, which will control which direction of heating and cooling. And the heat transfer will be controlled by the amount of power input into the thermoelectric coolers. We decided to use convection cooling because that will reduce that would create uniform heating and cooling throughout the system. Some advantages of our system is that it is symmetric in its design, which increases the ease of manufacturing, and it's easily removable from the structure, which will allow for modularity in the future. Next, please. This is our basic design for it. It consists of 6 fans, 2 heat sinks with the thermoelectric coolers placed in the middle, and all of them are placed into a case. One half of it is placed on the outside of the structure, and the other half is placed inside the structure for the heating and cooling. Next. So for our customer requirements, a big one was it has to be able to heat and cool between 4 degrees Celsius to 70 degrees Celsius. Our system is able to do it from about 0 degrees Celsius all the way up to 80, just for a factor safety. To heat and cool our subsystem at maximum temperature of 7 degrees Celsius, it will take about 500 watts of energy. And the overall cost of the subsystem is about $2,000. And to perform the closed loop control, we will have a temperature sensor placed near the culture plates, and it will constantly monitor the system. And like I said earlier, we will be using convection cooling to prevent condensations on the well plates, which will help reduce condensation, which is going to help with the sensing system that Brooke will be talking about next.
Okay, so the sensing system uses an M-Cherry filter cube set to measure the fluorescence intensity, and then a separate phototransistor to measure the optical density. By using the M-Cherry filter cube set, we're able to detect 53 different common fluorophores, and using a sensor backing, we're able to sense this from the top down rather than engulfing a culture plate from both the top and bottom. We gave this subsystem a technology readiness level of 3 overall because it has been validated analytically, but like the other subsystems, it hasn't been prototyped and validated in a lab. It's important to note that the off-the-shelf parts individually evaluated are at a technology readiness level of 9 because they have been prototyped and tested and are available on the market. But because they've never been in this configuration specifically before, the overall subsystem is lowered to a level of 3. So the system is mounted on a 15-degree bracket, which allows the laser diode to pass through the filter tube and be split down to the culture plate. And this way, the laser goes through each individual well at an angle, so there's no noise from the adjacent wells or the plate itself. And then using a mirror, it's bounced back to the PMT module, which both the PMT module and the phototransistor output a voltage, which can then be used to calculate the optical density and fluorescence intensity for the user. This overall system is mounted to the dynamics and translation system via the subsystem mount, which you can see on the top left over there. And with that, Ryan is going to be talking about the dynamic system. Oh, not yet. The customer requirements that are important for this subsystem is that the cost is about $2,300, which is 25% of the budget, but devices that are on the market that only do optical density and fluorescence intensity readings range about $8,000. So this is significantly cheaper than options available on the market right now. And another customer need was to design it with respect to trade-offs in monochromators versus filter cubes. And by using the M-Cherry filter cube set that we've chosen, we have a lower cost as well as a higher sensitivity than if we used a monochromator. Customer need 30 is the only one with a yellow key versus green, and that's because the customer need asks that we achieve a light intensity of 1 kilowatt at 640 nanometers. But through research, we found that using this intensity does slow microtubule growth within the cells by about 50%. And this is a indicator of phototoxicity within the cells. So to allow for a factor of safety while sensing things, we lowered the intensity to 600 watts at 640 nanometers, which provides a factor of safety about 6.67 and slows decreases the microtubule deceleration to 37.5%.
All right, so now moving into the dynamics and translation system. So the overall purpose of the dynamics and translation system is to provide transportation and motion primarily to the liquid handling system and the sensing system. The dynamics and translation system also includes the two shaking plates, which oscillate and gyrate the culture samples as they're being cultured. So a few key features of the dynamics and translation system: it's offering 3 axes of motion with X, Y, and Z motion capabilities. We're offering up to 3 unique shaking patterns with the 2 shaking plates. We're offering closed loop control for all motion systems with integrated limit switches, which act as a fail-safe for the system to prevent crashes and damage in the event of a programming error. The dynamics and translation system was given a technology readiness level of 2, since the system has not been prototyped yet. And like others have mentioned, all off-the-shelf solutions have been given a TRL value of 9. So moving into the details of the dynamics and translation system, we have it broken out into 3 main subsystems. So we have the X-axis assembly, the Y-axis assembly, and the shaking plate assembly, which you'll see on the next slide. So starting with the X-axis assembly, you can see that we're using a stepper motor for linear motion combined with a belt drive system. This linear motion system offers consistent positioning and reliability to move the sensing and liquid handling systems to where they need to be in the well plates. Similar with the Y-axis system, we're using a belt drive with a stepper motor attached. All linear motion components include bearings to provide rigidity and smooth motion, as well as consistent positioning accuracy. For the Z-axis movement, we're relying on pneumatic cylinders to give us Z-axis translation. This increases the speed of translation over something like a linear actuator, or for an electronically driven linear actuator, which resulted in a higher cost and slower response time than the pneumatic alternative. Moving into the shaking plate assembly, as I mentioned before, our shaking plates offer 3 unique shaking patterns. So we have a linear shaking pattern and circular oscillating shaking pattern, as well as a figure 8 shaking pattern. So starting with the linear shaking pattern, to generate this, we're using a ball screw and stepper motor combination system. So this is just giving us linear motion by moving the motor back and forth with the ball screw system. For the circular oscillations, we're relying on another motor drive system, which has what we're calling offset connectors to generate an offset motion to generate the shaking motion. And to create the 3rd shaking pattern, you're going to combine the linear motion as well with the circular motion to create that overall figure 8 motion that was the customer need requirement. Moving into the requirements for the system, we had 6 main requirements that we needed to meet. So the first requirement was the power consumption. So this system drew approximately 230 watts of power, which results in 30% of the overall power for the system. So it is a power heavy system that's generated from the motors as well as the valves, pneumatic valves. The life cycle must be 10 years. So the first failure point we determined was going to be the stepper motors. And through research, we determined that the stepper motors have an average life cycle of around 10 years, given normal operating conditions for 8 hours a day, 5 days a week. The total production cost for the system came in under approximately $2,200, which was 20% of the overall cost of the system. So that's right about where the other systems were as well. As I mentioned before, we generated 3 unique shaking patterns which met the customer needs through our shaking plate system. We also generated independent shaking environments for different well plates. So adding 2 shaking plates gave us full control for the 2 separate environments. So a little bit more about the shaking plate mounting. So the shaking plates have the ability to mount the custom well plate that Haley talked about earlier, plus 2 standard size well plates, as well as 12 conical, 15 milliliter conical tubes and 4 50 milliliter conical tubes. So the main goal with the shaking tables was to focus on modularity so we can create those unique environments as needed. So now we're going to move into the overall structure system.
So the main body of the overall structure will be made out of 10 gauge 5052 aluminum sheet metal. This body will be full seam welded and will feature 2 holes for access to the inside of the structure where most of the subsystems will be housed. If you look at the figure at the bottom of the screen, the first I will talk about is the hinge lid, which is the orange part at the top of the figure. The hinge lid will be utilized for assembling internal components as well as any sort of maintenance that is needed or deep sanitizations. This lid will be sealed with a O-ring and will be latched with a locking latch that you can see at the front of the structure. If we look at the bottom of the figure, the circular green plug we are deeming as the quick access port will be utilized for any sort of quick cleaning operations, as well as most importantly, inserting the cell culture samples. This design was inspired by our reverse engineering project, which featured a stopcock burette system. And essentially how it works is the plug has an O-ring around it, and the cylindrical extrusion in the front of the structure, the plug will be inserted into it and the O-ring will seal against the inside of the extrusion, similar to how the stopcock seals against the inside of the burette. The technology readiness level was given, for the structure, the technology readiness level was given as a 2. The reason for this is there has been no prototype made as well as no laboratory testing. So we need to validate our design before we were to increase the readiness level. And we expect that some minor changes may be needed once we are able to make a prototype. Next slide, please.
Yes, so on this slide, sorry.
Go ahead, Locke.
Okay, I'm sorry. So on this slide, you can see the callouts to major components on the structure subsystem. So now we did decide to not use an integrated display for data acquisition and control of all the components, such as a shaker table and the heating and cooling. So we decided to go with a standalone laptop because, as you guys probably know, as an example, cars with touchscreen 10 years ago looks very dated in modern time, like 10 years from then. As part of the customer requirements for the lifetime, the expected lifetime as part of the customer requirements is about 10 years. So we know that using an integrated display in 1 or 2 years, it will feel very slow and with high latency on the touchscreen. So the advantage of the laptop is that a scientist or a lab technician can just bring their own laptop, hook it up to the machine, check their code, check whatever program they want to run, the dispenser, heating and cooling cycle, and then take it back to their desk station to keep working. Now it does include a panel with LED indicators and an emergency stop. You guys are probably asking, if the box isn't see-through, as you can see, then what is the point of the emergency stop? Now we do plan to put a camera inside the structure assembly so you can monitor any zeroing movement of the liquid dispenser. And if you expect a crash is about to occur, you would hit that emergency button. Next slide, please. So here are the custom requirements we work with for the structure system. One of the main requirements we targeted was requirement number 4, which states that the system must be easily assembled and disassembled with minimum know-how. This is the primary reason why we decided to put a large access lid on top of the structure system instead of in the front. As an example, the dynamics and translation system weighs around 20 pounds. So it's easier for a technician to keep the CG of that component close to their body. So when they're installing it, they're going to hold the translation system closer to their body, then drop it straight down into structures because there's an open top lid access. Now, if that big opening was in the front, the technician would have to extend their arms about 24 inches, which is how wide the structure is. Now that is not an ergonomic installation position, and it's very easy to pinch your fingers and hurt yourself. Now, in terms of accessibility, we also have done tooling checks That means any fasteners or components that needed fastening inside the structure assembly, we actually put wrenches, torque wrenches, screwdrivers inside to make sure that the line of sight of the fasteners is not blocked by any other components that is not in the correct installation sequence. For example, a hex head fastener needs to be able to turn 60 degrees in one wrench cycle. It means if you can only wrench it like 10 degrees, it will be very annoying to tighten down a fastener. So those are what we focus on. And as Alec mentioned earlier in the first introductory slide, our 2 openings are sealed with O-rings, which I think is a unique feature for our system, because I've seen other groups and it appears that their openings are not sealed. Now, we don't want any contamination going inside the assembly, and we don't want any hot or cooled air leaking out. So that's why we chose O-ring seals for our openings. That's pretty much it for the system.
These are a couple of key features that we feel make our design stand out from others, such as the customizable well plates that Haley talked about. And we also have multiple access points, which allow the user to make changes from inside the design from different angles, such as the top and the side, which we feel really adds to the user friendliness of this design. This is an overall cost estimate of our design, listing parts, materials, manufacturing, assembly, and overall cost. And although we realize that the price is over budget, the main cost comes from purchased parts, and we feel that this is possible to bring down the cost from this section by selecting different distributors for the off-the-shelf parts. To conclude, Sabre is able to meet our customer requirements and provide an autonomous experience that is not currently available for its price point. And we would like to thank everyone so much for listening to our presentation today. We really appreciate all the support that we have received during this project, and we would also like to know if anyone has any further questions.
All right. So thanks, guys, for an excellent presentation. You did a great job. Let's open the floor to questions from the panel.
All right. I'm going to jump in here and say, fantastic job, guys. I think this is the first presentation that I've seen. So we're on slot 6. So we're a third of the way through. And the first presentation where I actually looked at systems and I went, yeah, that actually looks like something that I can build in Mech 3 and it doesn't make me want to slam my head against the desk. And I know this is being recorded and I'm perfectly fine saying that nonetheless. So good job, guys. Like it was really well done. It was a good presentation also, which I liked. Like you did very well there. The light intensity issue that you guys had, and this goes back to, I guess, what the customer needs are, and I want to make sure that I understand them also. Is that light intensity requirement for fluorescence intensity, or is that the light intensity requirement for doing white light for photosynthesis experiments?
I was under the impression that it was for the fluorescence intensity experiments because of the customer needs had a linked document with information regarding fluorescence intensity and the intensity of light.
So that wasn't the general, because it was like, there was one requirement in there, I think it was like maybe a nice thing to have, that was like broad spectrum white light. And that's specifically for photosynthesis. And I couldn't remember off the top of my head.
Right, we actually do have a white light for photosynthesis. We didn't mention it in this presentation, but it uses LED strip lights on the side that's closest to the user, so along the wall with the access port, and those are pointed at the custom culture plate to provide that light.
Cool. And I guess
is that, I mean, I don't know a whole lot about plants or LEDs, but I know photosynthesis tends to happen with mostly green light, and white LEDs are often sort of red, blue, and I guess green, right? So I mean, is the LED going to be sufficient to get the right spectrum of light for photosynthesis?
Yeah, actually, according to let me pull up my source, the Algae Research and Supply Group, they found that any typical white LED or fluorescent bulb can provide an adequate light to grow things. Because once you get too bright, the cultures actually die from it.
Cool.
I had a question about you showed the system set up with a laptop for control. Is the intent that however long this experiment is running, that somebody's going to have a laptop to connect to it?
No, sir. So if they want to monitor the data, they would leave the laptop connected, but they could also set up the experiment and take the laptop away if all they care about is the run status, which is indicated by the LED indicator. And once it's green, it means a cycle is running, some sort of heating, cooling cycle, or dispensing cycle.
So you've got some sort of onboard control that just connects to the laptop?
Yes, sir.
Okay.
Hey guys, great presentation. I really liked how thorough you guys were with the justifications. That's a really nice point. I did have a few questions for you guys in regards to, I guess we'll start with this slide. So you guys said that you're gonna later have like a camera put in there to better visualize what's happening within this like black box contraption. So that may be good for the user themselves, but if like we're doing one of these longer experiments and such where you may not be sitting in front of your, let's say your laptop, or you have it over an app on your phone kind of thing, is there some kind of like auditory signal to let people know who's maybe working in another part of the lab to like, once again, press the button if things are going awry kind of thing?
Yes, sir. So we plan to use a IoT, which stands for the Industry Internet of Things software called Splunk. So that will run and integrate all machine learning data within our system. So that we'll set up alerts using Splunk, and it'll email the users or whoever you approve as the user for this machine when a process has started, or when a cycle started, when it finished, if there's any error codes. So imagine like a heating and cooling cycle for when you're trying to culture these cells. I'm sure it's going to have some ramp up time, some dwell time at your heat, and then some cool down time or whatever. So now we'll have some sort of profile that the machine has to run at. If the temperature deviates from that profile, Splunk will notify the user saying, hello, sir, there's a problem. You might want to check it out. Then you would connect your laptop, or you could also stream the data, because Splunk is very good with machine learning data.
Oh, interesting. Never heard of that, but that sounds really awesome. I have another question in regards to sanitation of the overall structure itself. How's that being done with inside? Because I understand like at least you have that top lid, which I completely agree with your guys' ease of use of putting things in there. That makes a lot of sense, but how would you sanitize the environment after that point?
So we have UV lights around the system, UV strip lights that can serve as sanitizing the whole system, and also sanitize the waste before bringing the waste into the waste container.
Cool.
Wavelength.
Okay. Oh, sorry.
What wavelength are the UV lights at?
They are at 265 nanometers, which is the wavelength listed by the FDA as the most safe for human skin, so that when the user is cleaning the system, if there was some error and the lights were on, that would be the least damaging to a person.
So I'll give just a quick thing here. One, great job speccing that out. I don't think that's the best wavelength for sterilizing. Being that it's safe for skin, it means it's not going to be as toxic to cells, which you actually want it to be toxic to cells. The best way to do it is to put an interlock on your lid. So when the lid opens up, the LEDs, it's got a switch for the LEDs that'll disconnect the circuit for the LED lights. That would be a better safety approach there. But yeah, Robert, did you have more on the LEDs, on the UV to say?
No, but I do have more questions. So if we want to keep on that, we can.
To add about the sanitation as well is we focused on designing components with the ability to be autoclavable. So we found that generally autoclave temperatures run up to about 250 degrees Fahrenheit. So with the exception of some of the electronic components, we designed all the components to meet a minimum of 250 degrees Fahrenheit, so it could be autoclaved for sterilization.
Ooh. Okay, I had another question with your heating and cooling. Is that system located inside the system itself, or is it on the outside?
It's gonna, well, half of it's gonna be inside and half of it's gonna be outside because it has to constantly keep one side at a constant temperature. But it's gonna be heating and cooling the entire structure, not just the well plates.
Okay, so just within the system itself, I guess it brings up a point. Is the whole enclosure going to have this like the same atmospheric conditions kind of thing? Is that what your guys' idea with that is?
For temperature, it's gonna be the same for all of them, but for the gases, we were able to control each individual well.
Individual well. Wow, that's very impressive if that's what you guys were able to do.
So that's okay. Because my question here was why a custom culture plate? Because well plates have been standardized for like 100 years or something. Like they cost pennies per well plate. So why something custom? But if you're doing individual well by well gas control, there's my answer. And
yeah,
holy
because
shit.
I'm sorry, each individualized container has its own pipe feeding through it, through the right side of it.
Okay, so each well plate has individual control.
Yes, yes.
Not each individual well.
Yes.
Okay, I'm still impressed. I was just on the verge of being like dumbfoundedly impressed.
I guess I have another topic with those customized acrylic plates.